very much for having me today, and delighted to moderate the excellent talks for this session. We will start by having Dr. Mayank Verma talk about his work looking at strategies for adeno-associated virus-based gene therapy for APBD. Thank you very much.
Hi, everyone. Can you hear me?
Yes.
Perfect. All right. I am hoping you can see my screen. Okay. Are you able to see my screen or just the?
Yes. Yes. If you actually want to go up to where it says swap displays, there we go.
Perfect. All right. Thank you.
Yes, there we go.
All right. Hi, everyone. Thank you very much for allowing me to present here. It's been great to see some of the other work that's been going on in APBD. Especially want to thank the foundation for putting up these things. This, along with the biomarker conference that they recently put on, those are really helpful for people that are working in the community, or people like me that are just kind of getting started in these communities. So today I'll be talking about gene therapy, specifically adeno-associated gene therapy. Since this is an APBD foundation, conference, I don't need to do an introduction about APBD, but I did want to talk a little bit about what AAV or adeno-associated viruses are. It's a virus that was kind of described long ago in '65 and was really just thought it was a contaminant in a stock of adenovirus, which is a very popular kind of naturally occurring virus. You've all had adenovirus before. And it's sort of what we call a dependent virus. So it's incapable of replicating by itself, but it requires another virus to help it replicate. There's many different capsids with it, associated with it. So kind of the shell of the virus, there's many different types of it. And the shell determines what kind of tissue that this virus can infect. It is a somewhat common virus. So there's neutralizing antibodies found in about more than 35% of the population. Kind of the number changes depending on which area which part of the world you're on. The important part about adeno-associated virus for us is that you can actually manufacture this artificially. You can make a recombinant adeno-associated virus. So AAV has this natural packaging in the middle, and you can just take this out and put in another DNA element, things that you want artificially. Once you have this, you can package into different viruses, and there's different manufacturing technologies available to make the virus. Now, I just want to take a little bit of a pause and say there's actually there's a lot of different ways to do gene therapy. Common, especially with viruses, commonly, there's retroviruses, lentiviruses, just adenoviruses. And I'm going to highlight why we chose to use adenovirus. Adenovirus has a packaging capacity that is large enough for us to put in GBE, which is actually not really that much of an issue because it's a fairly small gene. It's sort of a low inflammatory virus, so the immune response to it is not necessarily super high. It is a virus that doesn't necessarily integrate into the genome as lenti and retrovirus do, which kind of there's issues of that having oncogenic properties, which is not necessarily a risk with AAV. And lastly, there's sort of a broad range of cells that AAV can transduce. One key thing to note is that that AAV doesn't necessarily integrate at a high efficiency. However, its transgene expression is dependent on this episomal quality. So, for example, when the cell gets transduced with AAV, there's a little kind of like a plasmid that hangs on. And so if you have a cell that's rapidly dividing, that actually gets diluted out. So that is one of the things to consider when you're picking your viral vector of choice. So when we're planning these things, our target tissue, say the neuron or the skeletal muscle or the heart, those are cells that don't necessarily divide very frequently. So if you have an episome, it'll stick around for a long time. All right. With that in mind, I kind of want to talk about our gene therapy program at UT Southwestern. Burton Nassian, who's one of the directors, along with Steve Gray. We have a large group of preclinical investigators, clinical investigators, support staff, and really kind of have a assembly line of gene therapy going on here. A lot of it is kind of focused on this intrathecal delivery of AAV via lumbar puncture. So many of you may know lumbar punctures, they stick a needle inside, between two spines. They can take a little fluid out, or you can also use it to deliver drugs. So why focus on this strategy? In this group, we have many people working on it, and just kind of want to give you one example of this. So this is AAV9 biodistribution after intrathecal lumbar puncture in a mouse. And you can see that this is staining for GFP. You can see that in the brain, it does get in a lot of places, and because it is injected kind of in the lumbar region, that expression is very high. What I'm gonna kind of point you towards is towards the right here on this side. You can see that there's a lot of transduction in the spinal cord, as well as the brain. And if you compare that to, say the liver, the spinal cord is still relatively high compared to that. For IV administration for AAV, for example, the liver is really just, orders, like 10, 20, 30 times higher than any of the other tissue. So this intrathecal technique is kind of a nice way to concentrate your virus into the brain. And another benefit of it is that we know now that it actually translates really well to other species. So a non-human primate, which is kind of a larger animal, there have been several papers that have been published that, IT intrathecal admission of AAV does still show wide transgene expression. And kind of the other really good point that I sort of touched on before is that because you're focusing only on the intrathecal space and the brain, for neurological diseases, you actually require much lower doses, in order of magnitude lower, which is more important from a manufacturing standpoint. While we can manufacture large amounts of virus, when you have to scale it up, it is kind of still a slow process. It's also a safety issue. As we know, there's been several deaths that have been associated with very high doses of AAV viruses. And so if you have the ability to kind of concentrate the virus into your target organs of interest, like the brain and spinal cord, then you can greatly reduce your risk of that. So APBD ends up being a good candidate for this AAV9 mediated gene therapy. First of all, it's a monogenic disease. There are several mutations, but all of the diseases are due to lack of activity. There's not necessarily a dominant negative feature that I know of, a mutation that I know of. The degree of disease progression is relatively slow, which does allow for intervention. Our target tissues, like the CNS, skeletal muscle, heart, have low turnover. And then for certain strategic reasons, we're really kind of focusing on spinal cord for our study, given the clinical features of the disease. For us, our vector design consists of this this kind of novel CBH promoter, which is an incredibly high, very strong promoter. It's sort of a Frankenstein promoter. It's got a CMV enhancer, a chicken beta actin promoter, and a hybrid intron that has 5' of a beta actin promoter and 3' of MLV promoter, MLV. And then we have a codon optimized human GBE one. And I should say, we are using the human gene in our mouse experiments because once we have this human gene, it's kind of we don't want to go through the hassle of going between mouse and doing the whole experiment and then being told by the FDA, well, this is not the same drug. So we are using the human gene in the mouse. And we've optimized assays to detect this human codon optimized gene product across different species that doesn't interfere with common infectious agents and can be work in mouse, rat, humans, and non-human primates. We've tested two lots of it and verified that we are able to package it efficiently. And when we first, the first experiment we did was we wanted to make sure that we could properly localize it. And so you can see here in just human cell lines that the protein goes where it should in the cytoplasm. And then we did a de-risking experiment. Of note, GB is very highly expressed in skeletal muscles. There's several neuromuscular forms of the diseases. So muscle was kind of a nice, easy tissue to do de-risking experiments. We injected the virus on one side of the muscle, not the other, at 1 month and waited 1 month, or injected at 3 months and then waited 1 month and looked for response there. We noted that even though in normal cells, there were no aggregates that were formed when you overexpress it, in the GBE1YS mouse, we did see that the protein localized to the polyglucosan bodies, where there were polyglucosan bodies, and where there were no polyglucosan bodies, it just went to the cytoplasm. So we were sure that we weren't making any aggregates. And then I just kind of want to give you a big zoomed out version of this. So this left one is treated with just PBS. This right one is treated with AAV. And just from grossly, you can tell that there's a big difference in the amount of polyglucosan bodies, PSD positive fibers that are there. So for kind of quantification, we did see a reduction, and this is kind of a tricky thing. Just remember that we are using right versus left. So we see less amount of polyglucosan body positive fibers on the treated side than the untreated side at the early age. And that difference is not as pronounced when treated at the later age. Another way to do this is to just biochemically look for insoluble glycogen, and we saw a difference in the mice that were treated at the early age, and we had 2 responders and 3 non-responders, and that was not significant at the later age. So we are that was kind of just a de-risking experiment, and we've started on this large preclinical studies. With more than 100 mice, where we're injecting this AAV9 at P10 at a low dose and a high dose. We have an interim time point for a certain biochemical analysis, and there we're going to be doing functional phenotyping and biomarkers for several times until we kind of end the experiment, hopefully when the mice kind of die by themselves. We have several functional outcomes that both us and others in the field have sort of seen some sensitivity to. And then one thing I want to touch on is we're also starting to look at biomarkers for APBD in this mouse, and we're kind of doing that concomitantly while we're doing this preclinical study. One such biomarker is GLUC4, which someone already has touched on that. So it's a glycogen breakdown product, and it's very commonly used in Pompe disease. And what's kind of been nice is that it's responsive to enzyme replacement therapy. So we know that when your kind of total load of glycogen goes down, this is supposedly would respond. So we developed this in-house as well. We have kind of two versions of it. One is kind of hex4 and gluc4. So to not get too into the details, the gluc4 is just kind of a more of a clear version of the hex4. It's kind of the more direct downstream that we're looking for. And you can see that in our sort of earlier version, when we looked at Pompe disease, we saw about like a 2 to 3-fold increase of the hex4 in the Pompe mice. And in our new version of the gluc4, we kind of see a 20 to 30-fold increase in the Pompe mice. With the older version, we also saw maybe like a 1.5, 1.6-fold increase in the GBE model mice. So now we're on our way to characterizing this with the gluc4 assay. We're hoping to see kind of wider differentiations between the normal and the diseased mice, and are really hoping to use this as a biomarker. All right. So in conclusion, we've kind of validated human GBE1 AAV9 virus with several in vitro and in vivo transfection assays. We now know that the GBE1 does localize to the polyglucosans when they exist, but otherwise it does kind of recapitulate normal expression throughout the cytoplasm. And then we've shown that the intramuscular treatment can reduce polyglucosan bodies following local delivery. And then we've started preclinical IND-enabling studies for using lumbar puncture intrathecal delivery of GBE. And along with these, we are exploring some translational biomarkers that could be hopefully used for trials as well. I just want to acknowledge the Manassian group, some of our funding sources, and other areas where we get a lot of resource and help from.
Thank you very much, Dr. Verma, for an excellent talk, and certainly as a resident physician, it's quite impressive work in that of your laboratory at UT Southwestern. And so with that, we'll introduce our next two speakers. We have from Columbia University, Dr. Orhan Akman and Dr. Michio Hirano will be presenting their work on amelioration of GBE1 deficiency by intronic mutation-specific antisense oligonucleotide therapy. And again, just a reminder to please enter any questions or comments into the chat box for discussion at the end of these talks.
Hi, everybody. Thank you to APBDRF and the organizing committee for giving us opportunity to present our research. So it's another great meeting that we come together and share our ideas and present our findings and look for other solutions and avenues that we can use to treat those diseases. Now I'm going to present my brief research history about finding intronic mutation in APBD patients. Because this, finally we are going to be able to at least develop some treatment that can be applied to a subgroup of APBD patients which were known as manifesting heterozygotes before we found this mutation. How do we advance the, okay. So what is an intron? The introns are the DNA sequences between the exons. So the name intron comes actually in the cell where the DNA is in the nucleus, and each chromosome contains many genes, and GB1 is one of them. And as you see here, the lines shows the whole GB1 sequence is more than 250,000 base pair. This is a huge number to analyze the gene sequence by sequence. At least it was 2015 or before. And so this is such a big DNA molecule, yet only 1% of it really codes for the protein that the cell needs, which is GB glycogen branching enzyme protein. And that all that information is in 1% of the gene. As you see here, all the exons has been marked on the DNA, and as the RNA is being synthesized, some signaling molecules, those are actually sequences of the DNA, marks the beginning of or end of the exons, and the enzymes removes this DNA part from the exons. And this whole exons brought together, and it gives the information for the synthesis of the protein. Now, of course, when we do the gene therapy, as Mayank showed before nicely, we don't use this DNA sequences in between. So most of the time this has been, or until recently, this has been known as a junk DNA, had nothing to do with actually the protein. So when it comes to the viral therapy, we can just easily get rid of it and use only the part of the DNA that codes the protein. However, these introns are in the genome, and of course it regulates, so now we know that they have many other functions. And of course, the changes in those regions affect the synthesis of the gene. Now, the APBD patients that have been described as manifesting heterozygotes, we did know that they were heterozygous, first of all, for the most common mutation, PY329S, as you see here in the electrophonogram, which shows the DNA sequence, and where the codon is, you see the two bands. One is belonging to the mutated allele, the other one is normal. And when we look at the messenger RNA, again, we see together, looks like it is heterozygous, but this is being showing this heterozygous because in most of the time, when we analyze the messenger RNA, we cannot amplify the whole mRNA, and we do part by part. And when you do part by part from exon 1 to 15, then you see this perfect heterozygous condition. So it clearly shows that, oh, the patients are heterozygous. So they are not supposed to manifest the disease, because we know that APBD is autosomal recessive disease. If you have one correct gene, it's not supposed to have the disease, yet those patients have the disease. After looking at the enzymatic activity, I noticed that the compound heterozygous patients or manifesting heterozygotes have even less branching enzyme activity than the homozygous patients. And of course, this prompted me to study even further and look at the whole messenger RNA from the last exon to the first exon. And when I target the amplification from the whole messenger RNA, I noticed that now the real messenger RNA that synthesizes the protein is actually homozygous for the mutation. Now, what happened? Why all of a sudden this perfect heterozygous condition become homozygous? I mean, this clearly indicates that there is something that excludes exon 16 from in the normal allele that doesn't have the PY329S mutation. And And of course, when there is no the last exon, the protein doesn't work, or our actually protein analysis show that it's not stable, it degrades, and that's why the patients have even less branching enzyme activity. Now, of course, like every molecular biologist, you think that there is something in the intronic region, or there's a big deletion. So the big deletion theory was not there in the first place, because we were able to amplify this region one by one, and and then it was clear. So we reanalyze the sequence using the poly-A as a reverse primer, and we noticed that there is some region in the right at the boundary of exon 15. We have some sequence that comes in addition to the normal sequence. This is the normal sequence. And when we look at the genomic DNA, we have this part which has been deleted, and this part of the DNA has been inserted in that intronic region. So that's why we call this indel mutation in intron 15. And this insertion contains a message. It's a message that actually, that I show you here, that tells the DNA to, okay, or RNA, okay, you remove this area or remove this area. But in our case, that area that has been removed, instead of this, it was this, and it included the part of the DNA which was mRNA, which was not supposed to be in the message to synthesize the protein. And this message was so perfect, it had a stop codon and poly-A site. So that's why the messenger RNA was so stable, and all of our analysis told us that, okay, the patient was supposed to be only heterozygous. Now, we know that this insertion contains a message for this aberrant mRNA synthesis, and it's also sequence specific. So we can actually target this area using antisense oligonucleotides and maybe neutralize this mutation. And if we neutralize this, where we are going to see this homozygous case, and here, supposed to appear like this one, right? So Ionis Pharmaceuticals help us to design the antisense oligonucleotides, and in the patient samples we treated with those antisense oligonucleotides that they send us. So it's a 19 base pair region. So they send us 23 oligonucleotides that actually spans this 19 base pair area. This is very crowded slide, but it shows basically the what messenger RNA looks like in each treatment, in each well. We treated with two different concentrations of antisense oligonucleotides, and we were hoping to disturb this structure so that next codon is going to be normal exon 16. So this is basically the more easy to see version of that. So if we don't treat the fibroblast with the specific oligonucleotides, as you see, we have the mutation. There is no normal band. And with the oligonucleotides, those are labeled according to their locations on the plate. So we start to see the normal band that appears where the mutation is supposed to be. Now, this clearly shows that we were able to change the messenger RNA synthesis and correct the other allele or neutralize this intronic mutation. And in one of those cell lines with the different concentrations of oligonucleotides show that we were able to rescue it up to a certain level, and later, I think it's toxic for the cell. It decreases the enzyme activity again. Now, we tested those the best performing oligos in peripheral blood lymphocytes from a patient we took and treat with this antisense oligonucleotides. And we were able to recover the enzyme activity almost fully. So in this particular experiment, the patient was actually heterozygous for this intronic mutation, but not for the PY329S. It is very unusual case, but again, it shows the 50% of the enzyme activity, and that enzyme activity was able to be recovered using antisense oligonucleotides. So this clearly shows that using antisense oligonucleotides can rescue the mutated allele, and it gives the 50% of the enzyme activity that patient needs. And in long run, of course, this correction may stop the disease or disease progression, or over time, we hope that it may also make the patients better. Now, of course, the next step is to bring this to clinical site and make those oligonucleotides synthesized for clinical grade and apply to the patients. And Dr. Minassian and the other, we all know that it's very expensive and it cannot be done without help. And we and Noran Foundation come to our help, and Dr. Hirano, who is now the physician who is in charge, and we are collaborating with Noran Foundation to synthesize antisense oligos for patient treatment. So, Dr. Hirano, Micho, are you online?
I'm here.
Yes.
Thank you, Orhan. Yeah, so if you could control the slides, and I'll just show just briefly 3 slides to explain what we've been doing in collaboration with the APPDF Research Foundation and with the N-Lorem Foundation. So N-Lorem is a independent foundation supported by Ionis, company that developed the antisense oligonucleotide therapies for spinal muscular atrophy and has been developing other antisense oligos. So they have the special expertise in antisense oligos. And so they've supported this N-Lorem Foundation, which is focused on creating personalized treatments for patients in the United States with what they call nano-rare diseases. That is, diseases that have fewer than 30 patients in the world with a particular disease or particular mutation. So these are so rare that no commercial company would ever invest money into development of the therapy. So this is really very generous of this foundation to support therapeutic development for these orphan diseases that would otherwise be neglected. And so Orhan actually reached out to N-Lorem first and explained his results, which he nicely presented, showing very encouraging results in culture fibroblasts of patients with this intronic mutation. And so with that information in hand, we spoke to N-Lorem, and maybe the next slide, I'll briefly go through what we've been doing to try to advance this antisense oligotherapy for the intronic mutation. So in January of last year, as I mentioned, Dr. Achman reported the initial results of the ASO treatment in the GBE1 mutant fibroblasts, and then Ionis recommended to Orhan to apply to N-Lorem. So I contacted Dr. Achman, and I contacted the APPD Research Foundation, and they shared with us a list of 5 patients with this particular mutation. So a few months later, I was able to evaluate 2 of the patients with this particular mutation. And we were able to use the information that we collected from these evaluations to submit the 2 first 2 applications to N-Lorem, which fortunately were accepted by this foundation. And in just this past January, we had a treating physician orientation kickoff meeting with the N-Lorem Foundation, and these cases are now cases number 19 and 1171. in their long lists of potential candidates for various treatments. Just in March, I evaluated a third patient and have submitted that application as well to NLORMS. Next. So that's where we are. The next steps forward is for NLORMS to independently assess the antisense oligos that ORHAN has already tested, and perhaps others, in their laboratory. So they want to independently validate the results. We've also been notified initially through the APPD Research Foundation that there's a fourth patient that would be a candidate followed at Northwestern University. And we've recently spoken to the clinicians involved with that case, and they may also apply to NLORMS, and jointly we hope that we'll be able to move forward with this therapy with NLORMS and the APD Research Foundation and these two academic institutions. We will need to develop a study design and a protocol to assess both safety and potential efficacy of the antisense oligos. This development will require a drafting and submission of an IND, investigator new drug application, to the FDA. This is a difficult process. If anyone has gone through this, it requires a lot of work to prepare information about the drug and its potential side effects and its chemical manufacturing control and so on. So hopefully we'll be able to do this together with the NLORMS Foundation and get through this bureaucratic hurdle, which is very important to explain to the FDA what the drug is and what the contents are and its potential safety and efficacy issues. Okay, next. And then finally, we'll have to submit and obtain approval for an institutional review board protocol at Columbia, and if Northwestern applies as well, we'll do it together with Northwestern. And so we've been through this journey for over a year now, and we've spoken to several people at NLORMS, including Sarah Glass, who's their Chief Development Officer, and Tracy Coles, who's their Senior Director of Research. And they've notified us that this process of from application to actually getting the SOs could take 1.5 to 2 years. So I just want to be realistic that this is not going to happen very quickly, but it will take time. But we're optimistic that we'll be able to continue to make progress, and we're very optimistic given ORHAN's very exciting results which he presented. So with that, I'll turn it over. ORHAN.
Thank you, Mitchell. And of course, I would like to thank APBDRF and KBS Foundation and our collaborators here, and Dr. DeMauro. So Jeff mentioned earlier that he helped at the beginning to found the foundation. So now I also would like to thank Ionis and NLORMS Foundation, of course. So if you have questions now, I can.
I think we'll save questions as a panel discussion at the end of the after everyone's talks. But overall, so incredibly impressive, the amount of collaboration, basic science, advocacy organization partnership, nonprofit organizations developing new meds, and all the way to clinical research. Really impressive work. And we will conclude our session today with Dr. Rebecca Koch's presentation coming from Duke University Medical Center on AAV-mediated GBE1 gene replacement therapy for GSD4 and APPD. Thank you.
Okay, do we see the right screen?
Yes,
Looks
looks
good.
good.
Okay. Perfect. Okay, yes, thank you. I am so excited to be here to talk on behalf of my research group. So I'm a clinical and translational researcher. I work with Dr. Priya Kishani's team, who's in collaboration with Dr. Badam Sung. And I was asked to speak on our lab's work on AAV-mediated GBE1 gene replacement therapy, which could be a potential treatment for glycogen storage disease type 4, including the adult onset form, adult polyglucosan body disease. So I'm simply acting as a messenger on our lab, but I'm excited nonetheless to be here to talk about some of our published work and expand a bit on our approach to AAV gene therapy for APBD. So I know we've had some background from other speakers, so I'll be quick with this part. But since we're talking about GBE, or I'm sorry, for gene therapy, I could not talk about GBE. So as you all know, we're talking about the glycogen branching enzyme, or GBE, which is encoded by the GBE1 gene. It's ubiquitously expressed with the highest levels, unsurprisingly, being in the liver and the muscle. GBE is responsible for catalyzing the last step in glycogen biosynthesis, and it cleaves that alpha-1,4 linked segment from the outer non-reducing end of the glucan chain, and it then transfers that cleaved oligosaccharide via an alpha-1,6 glucosidic linkage. So in patients that have biallelic pathogenic variants in GBE1, the result is a reduction or deficiency of GBE, which is what we refer to as a glycogen storage disease type 4 diagnosis. And this disease is inherited in an autosomal recessive manner. And rather than having that, spherical glycogen structure that we want, the reduction of GBE yields this insoluble, elongated glycogen chains that we refer to as polyglucosan or polyglucosan bodies. And those will accumulate in cells. And these polyglucosan bodies are excessively long with peripheral chains that lack that spherical structure, and it resembles that plant starch structure. So for those of you who are less aware of the spectrum of GSD4, I have a visual that kind of walks us through this incredibly variable presentation of GSD4. So I have the pleasure of working with both the pediatric GSD4 population as well as the adult population, so APBD. And to make matters a bit more confusing for all of you, the pediatric population is often referred to as GSD4 or Anderson disease, whereas the adult form is almost exclusively just referred to as APBD, and in many ways looked at as a separate disease, but of course all under the umbrella of GSD4. So we have children that are born with what we call this fatal perinatal or congenital neuromuscular subtype, where these neonates have this profound global muscle weakness and hypotonia, and unfortunately cannot breathe on their own and will die in early infancy. And then we also have children that suffer from progressive liver disease, and they can go on to develop liver failure and need a liver transplant. And then we have juveniles and adolescents who present with myopathy and progressive neuromuscular disease. And then we have, of course, the adult onset APBD, with most common complaints being neuropathic bladder, peripheral neuropathy, and gait alterations. So obviously here today, I'm focusing on the adult form APBD, which is at the far right of the spectrum. But because we're talking about gene therapy, I want to show the entire spectrum, given the variety of tissue involvement. And if there's one gene therapy for a subtype, it likely could be beneficial for some other subtypes, if not all other subtypes. So as we all know, there is a mouse model that recapitulates the APBD human condition, the Y329S mouse model. And so this variant is extremely common in the Ashkenazi Jewish population. Of course, the exact carrier rate is not known, but we know that it's incredibly common with 1 to 48 or 183 carrier rate. And there are, of course, pathogenic variants that have been discovered, outside of this Y329S, but I think we can all agree that this is the most prevalent in causing APBD. And so thanks to the work by Dr. Achman and his colleagues who characterized this model, we know that this mouse model has reduced GBE activity in the brain, heart, liver, and muscle, and that glycogen content is elevated in these tissues. And so I'll be referring to those tissues specifically throughout this talk, which is why I bring that up. And from here on out, just to keep things simple, I'll refer to this GBE1 YSYS mouse as the APBD mouse, just to, again, keep things simple. So using PAS staining, we're able to capture the progression of polyglucosan body accumulation in the APBD mice. So here, PAS staining shows the disease progression in skeletal muscle, liver, and brain of APBD mice. Please do not get overwhelmed by the writing. It was just more for those who prefer writing, you could read it there, but I'm going to verbally state it. So you can see the progressive accumulation of polyglucosan in the quadriceps, liver, and cerebrum. Although progressive liver disease is not typical in our APBD patients that carry that Y329S mutation, there is liver involvement in this mouse model. So just something for us to keep in mind as we talk about the impact of gene therapy. And not shown here is the heart, which seems to be less affected in this model, as only a few isolated clusters tend to have, PAS positive staining, so PAS positive myocytes, and those were observed at 3 and 9 months. So again, not shown here, but just to keep in the back of our minds. And I show you this because, again, I think it's important that we understand the model and how it could be applied with our therapies, and so we can, better understand the use of this potential treatment. So the topic of today's talk is a bit more on our AAV gene therapy work that we've worked on and continue to work on. So again, I'll be showing you the published results that were published a few years back that showed the systemic correction of, in this title GSD4, but it's in the APBD mouse model. So again, not to confuse you, but we are talking about that APBD mouse model. And I believe the speaker before gave a great overview of AAV gene therapy, so I won't go too much into the specifics, but we did choose to use AAV serotype 9, which has a high efficiency in delivering genes to the major affected tissues in this model, including the liver, muscle, and it can also pass that blood-brain barrier to get to the blood, or to get to the brain and the spinal cord. So human GBE cDNA was cloned onto that AAV vector containing a CMV-enhanced CV promoter, and then it was packaged as AAV9. And the AAV GBE was administered intravenously through the tail vein into 14-day-old APBD mice at 5e to the 11th vector genomes per mouse, so approximately 5e to the 13th vector genomes per kilogram. 6 treated mice were then sacrificed at 3 months old, so this first line right up here, after overnight fasting. And then separately for a long-term observation, an additional cohort of 8 mice were sacrificed at 9 months of age, so right down here, and again compared to those untreated. And then tissues were saved for histology as well as biochemical analysis. So separating out our cohorts by these that were stacked at 3 months versus 9 months, you can see the GBE activity here, so the actual activity of the enzyme. So this is an indirect assay, and it was measured, at both 3 and 9 months. And so if the treatment worked, we would expect the GBE activity to be elevated, if not near normal, compared to our wild-type mice. So in our untreated mice, you can see that it remains reduced, where you have 0.65% in the liver, 10% in the heart, 29% in the brain, 10% in the quadriceps, and 18% in the gastrocnemius, compared to our age-matched wild type. And so after treatment, so at AAV and right here as well, GBE activity was 26-fold higher than the wild type in the heart, so effectively transduced the heart, and moderately elevated in our skeletal muscles. It was 51, around 50% increased in the brain, which is approximately about 45% compared to wild type levels. But surprisingly, there was no significant increase in the liver. And I'll expand on that in just a few moments. And not shown here, the Western blot data correlate with this well. And at 9 months of age, activity was only seen in the heart. So down here on this lower level, it was only seen in the heart, and even that was approximately 71% less than seen at 3 months of age. So it's just something for us to keep in mind when we think about the long-term impact. So next, we want to understand if polyglucosan accumulation was still present after treatment. And so glycogen contents were then quantified at 3 months of age, which showed that skeletal muscle glycogen, so that insoluble glycogen, was similar to wild type levels in treated mice. Additionally, the liver glycogen was 59% of that in the fasted liver and 48% in the brain compared to these untreated mice. And so on the right, you'll see the PAS staining, which showed that AAV treated mice, again, had no glycogen deposits in the quadriceps and diaphragm, and they also had reduced glycogen in the liver and brain. So switching gears and looking at the 9 months of age cohort, the glycogen content was significantly reduced in the skeletal muscles, liver, and brain when compared to those that were untreated. And again, a note here for the heart, so right here in the middle, is that unfortunately the heart already has low levels of glycogen, making it difficult to assess, if the treatment worked in the heart. However, with PAS staining, which I did not show here, it again showed there were no visible glycogen particles in both treatment cohorts, so both at 3 months and 9 months. So staying with this 9-month cohort, we then stained for fibrosis using Mason's trichrome, and it revealed that there was mild fibrosis in untreated livers, so right here on the left, as well as it revealed the presence of foamy cells in the skeletal muscle of untreated mice. So that indicates that there could be some disruption of the muscle structure. And using immunohistochemistry, we saw that the foamy cells were positive for CD68, so for macrophages. And that wasn't shown here because I ran out of room, but just to show you that, we did see that it was positive for CD68. And so you can see with treatment that while GBE activity was not remarkably elevated in the liver, the gene therapy still reduced the amount of glycogen in the liver, and it protected it from that inflammatory cell infiltration, the fibrosis progression. And then on the right here, blood was also collected at sacrifice. And it was, again, after an overnight fast, and ALT and AST and CK were analyzed. And so you can see with the histology and the blood markers put together that treatment prevented that tissue disruption in the liver and muscle, and accordingly prevented that increase of ALT, AST, and then CK. So I don't have significance here because of the limited numbers, but again, I think we can see visually the decrease. And then for the viral vector count, which I think all of our gene therapy people are wondering, so RT-qPCR was used to quantitate the viral vector copy numbers in the tissues listed in this table. And so there is significant vector vector DNA in the liver, heart, cerebrum, and skeletal muscles. But at 9 months, so the second row right here, 90% of AAV genomes at 3 months were lost by 9 months in these APBD mice. So we have seen this waning effect before. It's been shown and published in the GSD1A mouse. So again, just something for us to think about from a holistic point of view when we think about the use of this therapy. So in summary, this was a proof of concept study for gene therapy in infant APBD mice. These results show us that the single injection of AAV GBE vector at a young age can effectively prevent glycogen accumulation in skeletal and cardiac muscles, and to a lesser extent in the liver and the brain up to 9 months of age. Again, this was intravenous injection, so I just want to point that out. AAV treatment also reduced plasma AST, ALT, and CK activities, and it did prevent the development of liver and muscle fibrosis. Now, again, this is a proof of concept study, but I think there's always room to grow and improve, and that's something we're actively working towards in our lab. So just some things to think about as we refine our approaches and think about the best ways to, best endpoints really for these studies. I didn't show it here, but a Rotarod test was used as a functional test, and there was no significant difference at 9 months of age, despite the reduction of polyglucose antibodies in the muscle and brain. So it suggests to us that, again, we need more sensitive functional tests. We also need to consider the use of biomarkers. I don't need to say that to this group, but just something for us to keep in mind as we think about markers of neurodegeneration and things that are more applicable to APBD and to some extent the neuromuscular form of GSD4 as well. And so we also need to consider in what way we want to utilize this therapy. So for our hepatic children and our neuromuscular children and adults, it's going to really direct the way that we inject. And I know that the speaker before me hinted at that, where, if we talk about intrathecal versus, intravenous, we have to understand the use of what way that we're actually injecting and what we're trying to target and how we're going to use at least this mouse model or other mouse models to direct that. And so I'm just ending here. I'm just very excited to share the work being presented at ASGCT next week by my colleague, Hai-Ching Li. He's also the co-first author on the publication from the data that I just described. And so this study tested an AAVF capsid to enhance the CNS transduction in young adult 6-week-old APBD mice. And this AAVF capsid is a new AAV9 variant. And so it's thought to be more highly capable of CNS transduction. And so this study design was using an AAV vector expressing mouse GBE. And so it's driven by a CB promoter that was packaged again as AAVF, intravenously injected into those 6-week-old mice at 2.5 × 10^13 vector genomes per kilogram. And then mice were euthanized 20 weeks later. And so this study has results that are very promising. And again, a proof of concept that if you successfully transduce the CNS, GBE activity can be increased and accumulation of polyglucose antibodies can be prevented in both the brain and the spinal cord. So I won't steal his thunder, but again, you can look out for this poster and you can also search it online for those who are not attending. So with that, I just have some acknowledgements. Thank you for my entire team. Really, I can't take credit for the work that they did, but also for the collaborators who made this work possible, for the APBD Research Foundation for allowing us to share our work, and of course, our patients and families. So that's it for me.
Well, thank you very much, Dr. Koch, for a very exciting presentation and certainly hot off the presses update. And thank you to all of our speakers, and we'll open it up for questions. We'll, some of the questions around n-Lorem came up, and I think it might be good just to give Dr. Hirano some time to talk a little bit about how do you submit this n-Lorem application? How is it different from a clinical trial as we know, and kind of where things are going forward?
Oh, okay. So as I mentioned, this is going to take time to set up this this trial. I would consider this more of like a compassionate use trial, but nevertheless, it is subject to many of the same regulations as a regular clinical trial. So there will be a requirement for an IND, probably a smaller IND, maybe an intermediate IND rather than a regular IND, it will require 18 months to 2 years to set this up. It will be challenging because although the company provides the critical reagent, the antisense oligo, we may need to have support for other aspects of the trial, including the patient assessments, any travel costs that may be associated with the trial, and regulatory work that needs to be done. So we're trying to piece all of this together. So it's going to be challenging, but it's very different from an industry-sponsored trial where everything is sponsored by the industry. Here we have to bootstrap everything ourselves, and hopefully we'll be able to do that through patching together some funding sources and whatever resources we can gather from the community.
Thank you. Dr. Kekwang, I don't know if you want to ask your question, or I'll ask it, whichever.
Yes, okay, I am whatever means. I just actually I was wondering if you can maybe comment on the difference between post-mitotic. Just discussed it with Dr. Rachman, but maybe we can open it for the panel. If you can comment on the difference between post-mitotic tissues and non-post-mitotic with regard to the effect of the efficacy of the 9 strategy.
Because of, APBD is basically, since polyglucosan accumulates in muscles and the liver, brain, and the tissues that doesn't divide like epithelial cells, so if you can give the gene with AAV9 treatment to those cells, technically they stay there as long as the cell is there. The dividing cells, as Mayank has shown, as the cell divides, the virus doesn't replicate with the cell, or the viral genome doesn't replicate with the cell. So the DNA dilutes as the cell divides. So that's why it is one thing. And if we can transfect the brain or the muscle cells, I think it is good to almost forever. Like in mouse studies, they never treat second times once the first treatment is successful.
So actually, this is why I'm asking, because for Andersen, it's basically a liver disorder. So what do you do there? I mean, for Andersen, for the neonatal or the juvenile forms, it's basically a liver disorder. So how can you address that? Repeated doses, or what do you do there?
Yeah, that is the downside of AAV treatment. Once you give it, you'll develop neutralizing antibodies against it. So currently it's not possible to do repeat doses, but there are strategies that people are using to enable you to do that. But that is a big kind of pitfall here. One thing to note though is that hepatocytes, can also be multinucleated. So sometimes you do see multinucleated. So in that sense, you may actually not be diluting it, but I think that's a small population that's going to be not very clinically relevant. It's just kind of biologically interesting. I don't think it's going to be clinically relevant.
If I can just add to, I think this is something that, I know other groups, when they're looking at OTC deficiency and Wilson's and other liver diseases, they've also considered with their clinical trials. And when we think about if we're just injecting into, like if you're hinting at again, like our classic progressive Anderson disease GSD4 patient, we also have to think about what their liver looks like when they would be injected, because a lot of times these patients, once we get a biopsy from them, they're extensive fibrosis, if not cirrhosis. And so, even as we're thinking about what patients are going to progress to needing a liver transplant versus not, I think it's also going to depend on what their livers look like, unfortunately, which I know even at ASGCT, it's a huge conversation about, because everyone's now afraid of injecting into liver disease patients, right? So I don't have an answer, but that was just another thought of something to consider for these patients as well.
So Dr. Koch, in your experience, what happens after liver transplant of babies? Is there relapse? What is your experience?
I'm sorry, say it again. Did you just ask about the relapse with the classic progressive patients?
Yes.
Okay. I mean, sorry, go ahead.
After the liver transplant, yes.
After liver transplant, depending on so there's a little bit of mix, but primarily the literature and in our experience too is once they have a liver transplant, it's not going to correct the extrahepatic manifestation. So if they do have any cardiac involvement or even neuromuscular involvement, it's not going to correct that. But typically that's not the lethal part, right? With these patients, we're talking about the liver failure. So if they are able to successfully get a liver transplant and post-transplant tolerate it, they are in essence cured of the liver disease part. But of course, it's not going to prevent the heart disease or cardiomyopathy if that's going to manifest, and same thing with any neuromuscular involvement. So it's not a perfect fix, but definitely it will keep them alive if we're able to get them a transplant.
And considering the liver, of course, if the liver is healthy, healthy means if it is functioning, like most of the AAV injected, it gets into the liver tissue anyway. And so dilution in liver is not going to be an issue. So because many copies of the viral genome is going to reside in the hepatocytes.
Okay, so dilution is not an issue. Okay.
So Dr. Verma, would you like to unmute to ask your question?
Yeah, it's one for Dr. Aikman. It's really great work on finding intronic mutations. That's a nice strategy and kind of hard to do. So once you find your initial mutation, how did you go about screening for these additional patients? Do you guys have a panel that you normally screen your undiagnosed APBD patients on, or kind of what's your pipeline?
Such a rare disease, and the intronic mutation is because of a founder effect. So like PY329S mutation. So we look at only these two mutations. Actually, we don't do it in the lab anymore. So it is clinically approved. So it is in most of the panels.
Okay.
And only thing is if there is any other mutation similar. Like I know that there is one case that has deep intronic mutation in exon 1, it's even more difficult because if it is at the end, the molecular biology allows you to amplify it. But if it is in exon 1, it is quite difficult because if you don't have the exon 1, you don't know where to start. And the first intron is like 50,000 base pairs. So that is kind of difficult. But to be able to know if those patients are really APBD or not, enzyme assay, and the best thing is actually do the biopsy and look at whole glucose antibodies. And mRNA study is important because I was able to find this mutation because we looked at the messenger RNA. And but even if you look at the messenger RNA, there has to be the other mutation that's going to tell you if the mRNA is homozygous, heterozygous, because if there was no PY329S mutations, I was not going to be able to find this intronic mutation. Those people don't even manifest the disease anyway. So So coming back
Orhan?
to your question, yes.
Yeah, it's so nice to see your work that led to identification of that mutation. As you remember, at the time, whole genome sequencing had just been introduced, and we applied that to your patients, and even that would not detect it at the time. You'll remind us in a minute why. I think it's a repetitive sequence, and the way the whole genome sequencing is traditionally done, it breaks down the genome into pieces and just sequences all the little pieces and bioinformatically puts it all together. So we couldn't find it with whole genome sequencing, and you had to find it with the elegant approach you described. But currently there's a long read, human genome sequencing that's possible. I feel deja vu all over again. At the time I advised you, Orhan, don't go into all this trouble. Let's just do whole genome sequencing, but it didn't work, and you had to go through all the trouble. For this new mutation you're looking for, maybe this time with whole long read sequencing, you can get it.
Possible, yeah. Messenger RNA sequencing is at least it's going to tell what is going to be, upstream or downstream of exon 2.
Yeah, but you can do long read sequencing on the genomic DNA. Once it's one piece of sequencing without cutting anything, and it should be able to show you. It's kind of expensive, but it's a few $1,000s. I mean, whoever is funding it, it's eminently doable.
If I may add, if I may add a comment, so Bears, you may remember we also tried in Hadassah the genome, and also didn't work like in your hands. But I was wondering, and now we also have a much higher depth. Would that affect? What do you think?
Yeah, the depth improvements would affect, but these days for complex intronic mutations, long read sequencing is the way to go, because it does not cut anything. It just, and finally the entire human genome has finally been sequenced. Like last month, there was a paper. Until last month, we had pretend completed the genome sequence, but now it truly is completed. So you can use these technologies.
We have a lot of interest in terms of how does one participate in a clinical trial. And I think the gap between animal model work through in human clinical trials is quite broad, but maybe each of our speakers can talk a little bit in terms of what they foresee as the next steps to getting to an in-human clinical trial.
Yeah, if I could kind of chime in. For AAV, at least the general pathway is there. There's several clinical trials, so there is somewhat of a pipeline to follow. As far as clinical trials in general, I think, this may be kind of my own opinion, but biomarkers are really going to be the limiting factor here for these like APPD. It's hard to imagine doing a 10-year trial where you can look for efficacy. So having reliable biomarkers that can show you progression of the disease and sort of something that's responsive to treatment, that I think is going to be probably the limiting step here.
I guess on the echo of the AAV part, I would agree with that. I was also going to say two thoughts. One, natural history, which I promise is not a plug for my natural history study. I genuinely, if you know me, I want to share the data, or at least the findings. So natural history will be incredibly important, I think, especially with GSD4, including APPD, because it's so incredibly broad. We really need to understand what we need to target. I think APPD, we have a general understanding for at least patients that are homozygous for that Y329S mutation, but there's a handful that are not, that we need to understand. And I know tomorrow we'll hear more from the patient perspective about symptoms that maybe patients experience that aren't as common. And the only other thing I was going to add to Dr. Vermo is just the route of administration. I think we hinted at it even when we were talking about the liver. Depending on what we're trying to target, it's going to be really dependent on the patient population. So again, with APPD, we're lucky because it tends to be more the symptoms are more well understood. At least we know what the symptoms are going to be for the most part, but how that will apply to related GBE1 diseases is, I think, up for discussion.
I want to say I agree with both comments about the natural history study being essential, because you need that as a foundation to plan your clinical trials. And then once you get to the trials, biomarkers are great and it'll take you to an accelerated approval, but that's not the same as a full approval. Ultimately, you do have to prove clinical efficacy in a way that's meaningful to the patients in terms of survival, function, or how they feel. So biomarkers are great, but ultimately you'll need to get full approval, you need clinical efficacy.
And Dr. Harano, not to put you on the spot, but
Yeah.
only individuals with the specific intronic mutation would be in trans or with another allele with a typical pathogenic variant in GBE1 would even be considered for the ASO that you had mentioned.
Yes, absolutely. And so that's why it's a very limited pool of patients. So they have to have that particular mutation. So it's a very small subgroup at this point.
We have one attendee who is interested in the generalizability of some of these interventions, namely if any of these might be helpful for other diseases associated with polyglucosan bodies.
I think for AAV GBE, we're out because that's only for GBE related diseases. So I don't really have anything to comment on for that.
Yes, so everything else, though, that was discussed earlier, targeting downregulating glycogen synthase in which way, should apply to most, if not all, glycogen storage diseases.
Are there any other comments or questions? Oh, Erin from the APPDRF, feel free to unmute.
Thank you, Dr. Abreu. And thank you so much, everyone, for the wonderful talks. I was wondering, and this question is probably mostly for Dr. Verma and Dr. Koch, are you aware of any specific challenges with regard to the translation of gene therapies to an adult patient population and specifically targeting the CNS? Dr. Verma, I know you spoke briefly about the potential of neutralizing antibodies. I don't know if you wanted to elaborate on that a little bit.
Yeah, so to kind of give someone a background, when you're doing AAV gene therapy, a lot of people, like a third to 2/3 of population worldwide has antibodies against that the virus, in which case it makes your efficacy goes way down. So one way to get around that is kind of doing the intrathecal, and that was kind of the strategic decision for us to do intrathecal. Is that even though the liver and the muscle are kind of big organs that we worry about, the spinal cord is really where we focus, and if we can get that quote unquote immune escape by just doing intrathecal delivery, then that would still be kind of a big benefit. As far as translation, there are already several clinical trials for intrathecal delivery, and for SMA2 now there is a phase 4 for intrathecal as well.
I guess the pro, if we're talking about benefits of an adult population versus pediatric, is at least for gene therapy, the FDA, for a safety perspective, would rather start with adults than pediatrics. So I guess for a pro, if we're talking with our families that are, children versus our adult population, that is, if we can show safety and efficacy in preclinical models of the adult model, it's a higher chance, at least for the adults, than for the pediatrics to actually be able to translate to the clinic or go to the clinic.
Thank you.
Dr. Murphy, feel free to unmute if you're on.
Hi. I just thought of, I've had a few single variants of uncertain significance reported from the INVITE leukodystrophy panel. And I'm not sure if they're looking for intronic mutations. So perhaps that could be a cohort of patients that might have them if they know how to look for them. I don't know. I haven't asked them if they're looking for intronic mutations. Just as a suggestion to find more.
Yeah, no, I think that would be a very interesting cohort to look at. But we have to ask Invitae if they're willing to share the information and the samples and how that would be done.
Yeah, I'll say having ordered the leukodystrophy panel number of times and it being sponsored, it potentially could be a resource to access, but it seems like it would have to be funded through some mechanism.
Yeah. Perhaps a faster ways. ORHED has an assay to measure GBE1 activity in blood, so that would be a better way to screen.
And I mean, if any biopsy has been done, that is the other.
Yeah, I was going to ask if they have I guess, Jen, if you're asking, I guess they already have the symptoms. And I don't know if they've had a biopsy that showed polyglucose antibodies. But I know that there's several groups that are doing more functional assessments as well to try and see, again, if they have GBE deficiency or reduction to show that in vitro.
Well, thank you all for your vibrant questions, responses, and participation in this session. And with that, we'll conclude our session on gene and antisense oligonucleotide therapy approaches for GBE1 deficient diseases. Thank you.